new. What I've done is I've cut on some of the parts of the lecture from last year and added a few new ones, and given it a different twist. I'm not going to be talking much about the early part of early visual pathways. I think Paul Martin and Jonathan Victor have covered that very extensively, I take it. So I'll be talking not much about V1, but mostly beyond, and what influences the higher areas have of V1. So now, as you know very well, the human brain, that's 2 kilograms here, but this is probably the, the best learning machine that we know. We do, of course, machine learning. We can do some wonderful things that our brains cannot do, but at the same time, our brains can do lots of things which the machine learning at the moment is not able to. Okay, now, much of this, what is able, there are various algorithmic tools the brain uses. Now, I don't know whether this inserts here is covering part of the slides. I have no idea. But I hope it doesn't.
We are seeing the slides.
You're seeing the slide fully. Okay, all right.
Yeah, because what you can see in I see
inserts here, which is covering part of my slide. I don't know how to get rid of that. Okay. Particularly the annex and a few things here. Maybe I can. I don't know how to get rid of them. Okay. Now there are various algorithmic tools that the brain uses. Some are the basic biophysics of the brain neurons and its supporting cells. There are action potentials, and you know quite a bit about that, and the various ion channels, which are all gated by the voltage and chemicals and even mechanical pressure and sound and all of that. And then there are, where is it? Okay. And to enable learning, you also have the HEP synapse. And here is a simple concept, but it helps to increase synaptic efficiency. There is long-term potentiation or long-term depression, which increases the efficiency of the synapse or reduces it. Okay? And there are various rules governing the connectivity between different areas. There are developmental rules, and also thermal connectivity also happens in the adult, but quite a bit of it happens during development. Okay? And how do the different areas communicate with each other? And one of the things I'll be talking about today is communication by coherence, by how neuronal oscillations between different parts of the area, different parts of the brain would help such communication. And then there's an attention network, which is an extremely important part of the brain, a part of the brain function, so that we are able to focus our processing at certain aspects of the environment and react to and respond to those things which are important for us. And then I'll also be talking about another concept called predictive coding, which essentially means that our perceptions, thoughts, and actions are all governed not so much by feed-forward inputs from the sensory world, but actually much of it arises as hypotheses, and the sensory signals are used mainly to update that, validate that, or veto that. Okay, I'll come to that later. And there are many other tricks that the brain uses to achieve its goals. I can't tabulate all of them. The main thing I'm going to concentrate are these things, these three aspects of brain function. Okay. Now, you might have seen this kind of connectivity before, where this is most of the visual areas, most part of the parts of the brain which are concerned with vision in some way or other. And there's a lot of many of these connections are going both ways, feed forward and feedback. The only thing that the retina itself in mammals doesn't receive any particular feedback, but otherwise all these areas, even the LGN, the lateral geniculate nucleus here, the first station, the thalamic nucleus here, gets feedback back from the cortex. Otherwise, these are all highly interconnected. Okay, so there is a lot of scope for top-down effects. Right? Now, first I want to sort of put to you a few problems that the brain that the brain is confronted by. One is in perception, it's called binding, but the binding problem, that is, every object has got its own, shape and size and color. It may be moving at a particular location, at a particular depth, right? And one thing we also know, when we go back to this picture like that, different parts of the brain are specialized to various extent for certain functions. The MT, for example, one part here, MT. I don't know whether you can see the arrows when I point out here. The area MT here is concerned with movement and also depth, and various other areas here, particularly in the what's called the infratemporal cortex here, concerned with actually identifying objects. And B4 here is concerned quite a bit with color perception. Right, okay, now, so there is a functional localization happening, right? Different areas are concerned with different things. And also another aspect of brain function is that if you look at the receptive field sizes, that is the area from where you can actually stimulate a cell in the visual parts of the brain, that's called the receptive field, their size increases. If you take V1, the early area here, the receptive fields are very small, many of them are small, very small, that you have to put the light in a particular small area there. And as you might already know, that many cells are orientation selective, they don't respond to diffuse light or spots of light, but we have to have a contour like an edge or a grating or whatever. Now, if you go to the next areas here, like V4 is somewhere there, and then the higher areas here in the infratemporal cortex, receptive fields have become larger and larger, but they also become more complex. Here there are areas, there are cells here which respond to your face or to your hand or to some complex objects, right? This is where object recognition occurs. In fact, these areas are destroyed. You have what's called like visual agnosia, where you're not able to identify objects. You may still be able to see lines and edges and all that, and you're not essentially blind, but you can't really identify objects. Okay, but they have a large receptive field. If you have a cell responding to your face, we can put the face in a quite a large area. The cell will still respond, right? And this is an important part of any object recognition system, because you don't want to have a cell which recognizes, say, a pen or a tumbler or a glass, which at every location you don't have enough cells for that, right? But so they have what's called position invariance. Doesn't matter where you put them, the cells still responds within the receptive field. Okay. Now, how do you solve this problem? So how do you put different attributes of the object together? Because color is present in one area and shape in a different area, movement in a different area. How do you put them all together? For example, if you have banana and an apple, you're able to identify the yellow color belongs to the banana and the red to the apple. How do you put these things together if they're happening in different areas? And also you lost information about actual location as well. All right. Now, there are different solutions to the binding problem. One is binding by synchrony. And then there's a feature integration theory, and it's called spotlight of attention. And Treisman suggested that earlier. And then various people have been working heavily on that. This is, so could both processes be involved? So I think should be two here, okay. Now, with regard to feature integration theory, spotlight of attention, I must say that this has been quite a powerful idea where we process only one thing at a time. For example, if you're looking for a face in a crowd, you don't process all those faces together. And in fact, the more faces there are, the more time it's going to take. It's a kind of a serial process, actually, right? We might process something like 20 or 30 faces in a second, but it's not as if you are looking at each face individually with your moving your eyes to them. That'll be too slow because you can only make about 3 eye movements in a second. But whereas we can go through 10 or 20 faces in a second very easily when you're looking at a crowd. So to solve this, one way that Attention Spotlight may be working is this. Say, for example, the information coming from the eyes goes to the thalamus and to the V1. And then we know that there are through different pathways. We already experiments in a number of animals and also human experiments are all suggesting that there are two basic streams here. One is a dorsal pathway, which is more concerned with where things are and leads to action. Suppose you want to reach out something and grab something, that's what dorsal pathway is involved in that. And so it's mainly concerned with where things are. Whereas the ventral pathway is concerned more with what the object is, right? And so that you perceive, and this, as I told you earlier, these areas are pretty important for perceiving objects and making sense of that. Okay, these two pathways are there, okay? Now, we also know that with various damage to the ventral area called what's called agnosia, people are able to perceive things, whereas if there is a lesion to the dorsal area, that leads to difficulties in goal-directed action. You're unable to reach out things and do things appropriately. Okay. All right. Now, I'm going to build up a few issues here so that to show how these issues can be solved. Now, there are various different visual areas here, and you suppose you flash something, you know, some pattern or object on the screen, and this shows the latencies when the cells respond. These are dorsal stream areas here, right? They are somewhat faster, right? These input to the, I should tell you here, these inputs to the dorsal area, they're carried by a different system of neurons called the magnocellular pathways, which have got a faster conduction velocity, right? Whereas much of the ventral areas here are driven by parvocellular system, which is somewhat slower conduction velocity. It also gets magno input, but the most of the parvo input goes there into the ventral stream areas. They are these ones here in green. Okay, you can see these areas where object recognition occurs, they are somewhat have a longer latency. It's about 150 milliseconds or so, all right? Whereas these are much faster. Now, another, I'll bring you another issue here because I want to build up some of the problems we have and then how we might solve this. And this is, a simplified version of the visual pathway. The retina input goes to that genicular nucleus, it's one of the main way stations on the way to the cortex, and then it goes to primary visual cortex, and then it goes to two stream, dorsal and ventral, okay? And there's an alternative pathway as well, superior colliculus, pulvinar, and to the parietal cortex here. Now, the problem is, if you have a primary visual cortical lesion is damaged, the person is effectively blind in the sense that they can't see anything, right? They don't report seeing anything. But you can do some forced choice situations where you ask them to point to a light and going to flash somewhere. They get much better than chance. Even though they are not aware of the light, they say, I'm just guessing. I don't know. I don't see anything, but I'm just guessing, right? So which means that the information does somehow get there, particularly the dorsal stream, which is what I told you, which is necessary for goal-directed action. They're able to actually point to the place where the light is shown, even though they're not aware of the light itself, okay? Which means this is what's called blind sight. They still see, even though they're actually aware of the object. Okay. Now, it seems there are alternative pathways which take the information to the dorsal stream through that, and there are also some fibers going from the geniculate to the parietal cortex to the dorsal stream. Okay. Now, these are mediated by these inputs to area MT, which is an area in the dorsal stream here, which projects to posterior parietal cortex, right? The MT gets input directly from the LGN, right? So this must be responsible for that. But the ventral stream doesn't have any inputs that go there bypassing the primary visual cortex. This effectively everything has to go to the primary visual cortex. So we thought that you are not aware of things, right? You are not aware of the object. Okay. But at the same time, even though the ventral region here, the inferotemporal regions are necessary for object perception. Now, if you have parietal lesions, also lead to perceptual problems, what's called hemispatial neglect. In fact, if you have a lesion in the right parietal cortex, that is again in the dorsal area here on the right side, if you have a lesion, you have a neglect of one half of the body. So these people may neglect their left half. And in fact, if you put a plate of food in front of them, they eat only one half, right? So they don't see the left part of the plate, but they will complain what's wrong, that they're not being given enough food. And you turn the plate of 180 degrees, then they will eat the rest of it, right? So and they don't really attend to their own body on one half. It's called hemispatial neglect. And they may bump into objects on the left side, right? Okay, so it appears that the parietal cortex is necessary for perception, even though we said earlier that the recognition of objects and awareness of them does happen in the ventral stream. Okay, and also in other experiments where neurosurgeons have done small microstimulation before tumor removal, because the brain itself doesn't have any pain receptors, so you could have actually a patient conscious, and then you can put electrode and stimulate the areas, and it's necessary often to do that, because when you remove a tumor, you should remove enough of the new tumor, but not too much, and not areas which are vitally important, such as the language area or something. So these experiments show that to be consciously aware of your motor actions, that is, they're asked to do some motor action, or they're asked to intend emotion. When you make the intentions, it seems to be important that you can make this small micro stimulation in different areas, and that causes conscious intention of making a movement, even though they don't make the movement. They think they're making the movement. If these stimulations are made in the parietal cortex, right, certain areas, they feel they're actually making a movement, even though they're not doing it. At the same time, if you make a stimulation in some other areas in the premotor cortex, they might actually make a movement, but they're not aware of it. It appears that the parietal activity seems to be important for movement execution and movement awareness, okay? Now, all this leads to a problem, and there are some more things here I want to point out to you, that some experiments done by Benjamin Libet in the 1970s showed that you need to, if you stimulate the cortex like that in a conscious patient during a neurosurgery, you need to stimulate an area for at least half a second to be aware of the sensation for which the particular area is responsible for. For example, in the somatosensory cortex, if you stimulate, you will feel the sensation in some part of your body, in the hand or fingers, or the face or whatever. But you have to stimulate it long enough to be aware of that, right? And also, another interesting thing is, very often, your cortical activity, which actually leads to a particular decision, right, happens quite a while before your actual awareness of making the decision. I haven't shown those experiments, but these are other experiments we know about. And as I told you just now earlier, the damage to the ventral stream, not the dorsal stream, causes agnosia, right? Dorsal stream lesions actually cause problems with regard to actual actions, like reaching out to something or grasping something, okay? And I also, just as I told you earlier, right, you have this blind site where dorsal stream input that bypasses V1 is able to is processed somehow, even though one is not aware of that. And as I told you earlier, that normal parietal activity is needed for awareness as well. Okay, now one way of putting all this together, one solution for this is simply that that's where attention comes in, that you have this input, a fast input, and there are different inputs going to the primary visual cortex, a fast input and a slow input, actually different pathways. The fast input is the one that drives the dorsal stream, right? And that input can reach the parietal cortex here and the frontoparietal network here, now before the slow input even reaches the, arrives at B1. So when the slow input arrives in B1, suppose there are 3 different objects there, and you want to attempt to process each of them, but as I told you earlier, there could be confusion here that which later on you have the binding problem, which one may be a banana, one may be an apple, and one may be a pear. And how do you put all these colors and shapes together? But if you were to process only one at a time, right, the problem can be solved. So you can pick up one of those locations. The dorsal stream dorsal stream codes locations very well, even though it not be recognizing objects very well. It's the concern with where things are, right? So it may pick up one of those things, and one object is then processed. It picked up, and only that area is facilitated. So you have the inputs coming in from all these different objects, but the dorsal stream then feedback picks up one of those locations, and that location is facilitated, and others are sort of inhibited, and only that gets processed. If only one object gets processed, you no longer have a binding problem, okay? You see, processing one after is a serial process. And then if it's not the object you're looking for, you can go to the next one, okay? Okay. So there is a neural substrate for dorsal stream areas to actually gate and modulate what goes in the ventral stream, okay? This has been suggested by various people, okay? Now, what's the evidence for all of this? One way of this, many people have recorded from various areas of the macaque brain, from the primary visual cortex and various parts of the ventral stream, various parts of the dorsal stream. And in fact, in many of these areas, one could see, oh, this is from the last year, I think. Many of these areas show, almost all these areas show responses which are modulated by attention, depending upon where attention is focused. This is the spotlight of attention, right, which focuses on one input coming from one object or one location at a time, right? Its activity can be seen in many of these areas. For example, this is a study done in human subjects where we have if you do functional magnetic resonance imaging, I'm sure you all are aware of that. We can light up different parts of the brain, if you were to fixate at one point and show some objects at different distances from the fovea. The fovea is the central part where you have the maximum visual acuity, right? At this sector, this different parts of this particular sector, the activity in the brain also can be seen to be moving across like that. When you do it very close to fovea, just here alone, right? Here, right at the back here, at the posterior part of the brain, just at the back here, that gets stimulated nicely. That's the maximum activity there, seen here. And as you move across, the activity actually moves across here, right? This is when you were showing only that particular object here. Only that part is stimulated, okay? Now, there's another interesting experiment. This is nothing unexpected, right? In a basic feed-forward system, without any feedback, this is what one would expect, okay? Because there's only one region being stimulated in the visual field. And naturally, you get the stimulation there, the cortex. But the next thing they did, an interesting thing, they got all this stimuli here all the time. All the subject needed to do was to focus their attention on one sector or another sector here, another segment here, one segment or another segment like that, okay? Now, when they did that, even though all these areas here, such as here, all of them were receiving visual stimulus, visual input, right? Then as the attention was being focused on different segments here, the actual activity moved across the brain, just like as if you are actually having the segment, only that particular segment out there, as in this case. This shows there's kind of an attentional spotlight actually coming from higher areas which is actually increase in the activity in those areas here. Okay. Now, this is what we find with humans. But before that, a couple of years before that, I was doing some experiments back in Canberra those days, where the monkeys were trained to fix it on a spot. A number of stimuli, a number of gratings that they flashed. They were then had to focus their attention on, say, the receptive field of the cell was here, and these things gets flagged. This is in V1, primary visual cortex, okay? And the monkey had to respond and say this is a vertical or horizontal grating by releasing this lever. So the monkey has a lever. It presses the lever, fixation spot comes on, stays for a while. During that time, the gratings get flashed briefly, right? And one of those gratings gets a small little a cue about that. And the monkey has to attend to that grating and not to any of the others and say whether it's vertical or horizontal, okay? If it's vertical, it releases the bar when the fixation spot in the center dims. If it's horizontal, he has to wait until the fixation and the dim spot disappears entirely. Then the release is done, okay? Now, if you do that, so sometimes the cue will appear here, sometimes cue may appear somewhere else, such as there, in which case, in which case the monkey has to attend there, but not here. So you can compare responses of this particular neuron V1, the receptive field is here. In those trials, when the monkey had to attend to this grating and say with its vertical or horizontal, compare that with those trials where the same grating will be falling here, but it had to attend to a different location, okay? Right, when you did that, you find these are 6 different neurons. The difference there are quite dramatic differences in many cells. I'll pick up one of those cells now here. The monkey was paying attention to the particular grating to it, but it was cued to that particular grating. You get responses, much better responses than when the monkey had to pay attention to different grating. Even though in both these cases, the stimulus is exactly the same on the receptive field, there are different responses here, okay? Some cases, the other case, the response, was there. In other cases, the response is quite prolonged here, quite sustained response you get when the monkey is paying attention. Even though the stimulus is gone, the neuron is still responding quite nicely for a while, if the monkey had paid attention to that particular location, okay? Now, when monkey is paying attention elsewhere, you don't have that. Just when the stimulus appears there, you get a brief response, that's all, but you don't get this prolonged response, okay? And also, this kind of increased response comes much later, not early part. This stays for about 100 milliseconds, I think, or 50. Now, you find that in the first 50 to 100 millisecond window, there is no difference whether the monkey is paying attention to that location or not. But in the 100 to 200 millisecond window after the onset of the stimulus, you find quite significant differences between the two. When the monkey is paying attention, the response is much better than when the monkey is paying attention to different location. Okay, so even in V1, an early part of the visual path, you find this attention modulation. Okay, now we have tried to. Excuse me. These are experiments done with Yuri Salman, who is now in Wisconsin, and Ivan Pigarev, who is come from Moscow. We did experiments here in Melbourne. Here the monkey is trained on what's called delayed multi-sample task, right? Where the monkey presses the bar, the fixation spot comes on, and there's one grating gets flashed after a delay period of 280 milliseconds. There's no grating. Another grating gets flashed. The monkey has to compare both its orientation, whether the same orientation as the original one, and also the same location. It's a match trial, when a second grating falls in the same location and the same orientation as the first. Okay? In which case, what he has to do is that, so if it's a match case, he has to wait for the spot to dim. As soon as it dims, he has to release the bar and he gets his juice. And if it is a non-match, as in this case, same location but different orientation, or same orientation but a different location, or they're both different. In all these non-match cases, he has to wait for the spot, for the dim spot to disappear entirely, then release the bar. Essentially, he is looking there and doing a delayed multi-sample task here at this location. So he has to pay attention to that particular location, wherever the first grading. He doesn't know where the first grading is going to appear. It could be anywhere. But once the grading has appeared, his attention has to be focused on that place to see what the second grading is. Okay. Now, this case we recorded is from two areas simultaneously. One is area LIP here in the dorsal stream, another from MT. The inputs from V1 go to MT. From the MT, they go to LIP, and there are feedback from the back to MT and also direct feedback to V1. Now, so we record from these two areas simultaneously to see whether there's any feedback coming back from LIP to modulate what's happening in MT. Okay, so we had these two electrodes, one here, and we also need to match them for the same stimulus location, for orientation and for location. So the receptive fields had to be looking at the same place in the visual field, and they should be matched to the same orientation. And when we do that, and we record it, see, we found some interesting results. One is, so there are two gradings getting flashed. First, let's call the first one S1. It falls, say it's fallen in the receptive, in some trials, it will fall in the receptive field, right? Fixation spot is there, and it falls in the receptive field, okay? Now, there are, so fixations, there's the fixation period here, and there are one stimulus and the second stimulus, each lasting for 100 milliseconds. There's a delay of 800 milliseconds. And if it's a match case, the monkey has to release the bar during the period when it is dimmed, fixation spot is dimmed, or if it's a non-match case, he has to release it after the spot disappears. Okay? Now, first grading appears, we call the neutral condition because the monkey's attention has not been captured by any one particular location because that grading could appear anywhere. Okay? But now, let's look at these. The second grading, there are three possibilities. One, it could fall on the same location here, right? The same location. Yeah, okay. This is S2. We are looking at now trials where the S2 is always the same, that is falling the optimum orientation, falling on the receptive field. But some trials, the first grading would have been the same, the S1 would have been the same orientation and falling on the receptive field. Some trials, the S1 would have fallen on the same location, but a different orientation. And in some cases, it will be totally different location, okay? You look at how the responses to these three cases are, where the second stimulus is the same for all these kinds of trials, but the history is different. First grading, where it was and what it was different. If you look at that, you find that this is the case, match case, where the first and second grading, they both fall in the receptive field and they have the optimum orientation, right? They have the red traces here. And this is the case where the green cases are there where the first grading and second grading, they both fall in the receptive field, but the first grading is the non-optimum orientation for the particular cell. The response is less, as you can see here, right? Less than the optimum orientation. And also you see the population response. These are response of a single neuron in MT. The responses of the whole population is again a response of a single neuron LIP. LIP is the parietal cortex, right? And here is the population response. You can see that the first grading response is less because it's the non-optimum orientation. And the blue trace is when the first grading has been somewhere else, so there is no response at all for the first grading at this location. Okay. Now, you find that both in MT and LIP, the increased response for the second grading compared to the first, even when they are both identical and matched, increased. This is called attentional enhancement. This is nothing new. This has been shown to a lot of people. And irrespective of whether in MT and in LIP you find this increased response, irrespective of whether the first grading was this optimum or non-optimum, you still find increased response here, okay? And it's somewhat less in LIP than in MT, but in MT, they're both almost equal. And interestingly, in LIP, which is a higher area, which is supposedly sending feedback to MT, you find a ramping up here before the second gradient there, okay? Slowly increasing response. It's as if LIP is getting prepared because attention has been captured by this one. And somehow in the brain, it has to now look, we're attending to stimulus happening on that particular location to see what is going to be the next one. So the LIP is already having increased response there. Whereas if the first grading has been elsewhere, these neurons don't show that. Neurons in that responsible for the other location would be showing that, okay? Right. And then since we're recording the two areas simultaneously, we can also look at what's called coherence, how much these two areas are in synchrony. We used the programs developed by the multi-taper spectral analysis developed by Partha Mitra and his colleagues, or Chronex. And we found that when the monkey is paying attention, monkeys in both these cases, whether it's red or green, you find that there's an increased coherence during that time, which means in the particular frequency band, this is time here in the first grading and the second grading, and the different frequency bands here. And you find neuronal synchrony is higher. That is, when you say frequency and oscillation, the neurons simply don't respond sort of randomly. They have particular frequency, the particular frequencies, right? And then it could be faster, slower, right? And you can see it in local field potentials where you're looking at groups of neurons responding, or also on single units. And you find that you find a greater coherence between MT and LIP during this period here, when the monkey's attention is captured, but not for the first one. When the first grading appears, right, the monkey doesn't know where it's going to be because attention is not particularly focused in a particular location. But after that, by the time the second grading appears, even before that, there is greater coherence between MT and LIP. Okay, and whether the first grading is the same or not, right? The spatial attention, once attention has been captured by that location, right, there's increased coherence between MT and LIP. On the other hand, the first grading has been elsewhere, you don't see that coherence at all. I mean, the coherence will be happening in that other location, on the MT and LIP areas, which are looking at the other location, okay, not this location. Okay. Now, it's more or less showing all three. Probably we'll leave that slide. And you can also see, this is actually looking at single spikes. You find the same thing. I'll probably leave this too. Now, actually, you can see in single trials, you can see this increased activity here, MT and LIP, right? You have this gamma frequency here. You see the increased LFP here. Activity you can see. Right? Every trial is more than here, right? And you don't even need to do the averaging. You can see it in singles, naked eye. But you don't see that when the attention is somewhere else. And the attention in that location, if there is increased activity here, the attention is elsewhere. During the S2, this S2 is here, you don't find this increased gamma activity. And if you look at it a bit more carefully here, right, you could look at the spikes in that location here. This is a local field potential, right? These are actually individual spikes. We have the MT spikes and the LIP spikes. We can see this kind of grouping of activity, right? Now, but the MT spikes seem to be preceded by LIP spikes, right? Particularly in this case, in spatial attention. When the attention is away, it's not quite the case, okay? Now, so it could well be that the LIP is actually driving these MT neurons. And you can put that in a cartoon here. And LIP cells and MT cells, they are probably asynchronous initially. They're not in synchrony, but once spatial attention is captured, they seem to be in sync. LIP neurons are in sync, and say these spikes occur together like that, and they oscillate together. And they seem to be driving MT neurons also. They also fall into sync here, right? And which means, if you look at the, say, membrane potential, you can look at the membrane potential as going and oscillating like that, creating these spikes which come on top of this here when it crosses the threshold, membrane potential threshold, a threshold from spike initiation in the membrane potential. Now, you can imagine when the membrane potential, if you were to remain in the resting membrane potential, you'll have to raise the whole thing all the way up, right? But on the other hand, if you make it oscillating, any input coming in, next input coming in here, that's for the second stimulus, it can ride on the top of these peaks and reach threshold quite easily. Right? That is what probably is doing. So this kind of neural oscillation is a fundamental mechanism for one cortical area driving another one. Okay, so it's as if if you want to push start a car, is better, and the 4 people are push starting the car, it's better all 4 of them do it together, right? That's why you have the synchrony here, right? And instead of each one doing separately, so it gives increased, the synchrony helps it, and also this kind of oscillation helps it to reach the threshold for spike initiation much more easily. Okay. Now, going back, now we're talking about top-down effects, and now you can see predictive effects also in V1. I don't know how to remove. Oh yeah, I can move this around. Okay. All right. Oh, I don't know whether you guys see it. I'm trying to move these insets away. So these are 3 different cells in V1. It's actually in V1. It's an early experiment we did, where you see these 2 identical match trials, the identical stimuli, first and second. But you can see how different they are when the monkey is paying attention to them. The monkey is also is trained in this active task, which is the task I was telling you about. But there is also monkey also in another block of trials. It doesn't need to attend to the stimulus. It just looks at the fixation spot. We make the fixation spot slightly reddish. So when the reddish fixation spot comes on, he simply needs to fixate at the spot and release the bar as soon as the fixation spot disappears. So that's we call the passive task. Now, the passive task, in this case, there's no response at all. In these cases, the response is the same for the first and second stimuli. But in the active task, there's a considerable difference in the responses. Okay. Now, many cells in V1 are like that. So, which means some of the top-down attentional responses, attentional influences come right down to V1, not just stopping with empty, but it's coming down to V1 to influence responses here in some way. And this is an experiment also done along with Jay Kumar, who is there, Jay, and where we actually did microstimulation LIP, right? And we could influence the monkey's responses, the reaction time. This is actually reaction times here. If only if stimuli are presented at the receptive field location, right? Cells near the electrode. And if the stimuli is presented elsewhere, right? There is no effect of microstimulation. But microstimulation is made. Actually, you stimulate the cells a bit more, and the reaction times get affected, right? Okay. So these red ones are the microstimulation. Here, the reaction time is less, or the response to things faster. Okay. Now, I just want to briefly mention about some other things in LIP. LIP cells are also formulated by 2 groups of cells there. There are some cells which show this attentional enhancement is more response for the second stimulus, right? But these cells often do not show, these cells which show considerable significant attentional enhancement for the second stimulus, because the monkey's attention has been captured by that location, often do not show much orientation selectivity. Therefore, see the difference between, difference is not great. See the response, vertical grading, the horizontal difference is not great. But there are other cells which there is not much attention enhancement for the second stimulus, like in here, but those cells show often very significant feature selectivity, right? They have very different responses. Now, so this, from that, we be different for that, and we find that these 2 activity from this we can, how we look at it is that it gets some sort of feature or input is get. Suppose you are, there are 2, in attention you require 2 stages there. That is, if you're looking for your car, you lost your car somewhere, you've forgotten where you parked your car, you use a red car. So what you would do initially is to pick up all the red cars and then look which red car is yours. So the initial parallel stage, which picks up, all right, number of objects on the base of feature, and then you look at each one, so that the one I told you earlier, the spotlight of attention, comes on in the second stage. With the serial stage, the spotlight of attention is focused on one after the other. So it well could be that the initially Dawson stream, based upon it does both some feature selectivity, which is used in some way to cover a map of saliency, a map of locations where it need to attend to one by one after the other. And then a second stage where it actually focus attention, okay? I wouldn't go through this, but I'll show you this one where we were able to isolate these two types of activity, the feature-related coherence between LIP and MT, because they seem to happen at higher frequencies, higher in the gamma frequencies from about 40 to 150 or so, whereas the attention-related activity, feature selectivity, happens much below. It's actually only between 20 and 50 hertz. But these two kinds of activities seem to be quite different, kind of a different coherence between LIP and MT. The feature-related one, right, happens earlier. In fact, after the first stimulus is gone, that means during this piece probably retain some kind of memory where the locations are, where all the important locations, where all the red cars are they kept somewhere active in LIP. And then the second stage, where you throw attention, that comes much later, when you need to actually attend to the second stimulus and respond to that appropriately. So we seem to find this kind of difference between the two. Now, before I finish, this is the model we created, talk about that now. Finally, I want to briefly talk about predictive coding, what I said earlier, that most of the perceptions and thoughts and feelings and everything, probably one idea is that it's not driven simply by some feed-forward signals which create all these things, but probably they are called perceptual hypotheses, that we see effectively you have some random processes, like you can even compare it to mutations in evolution or whatever, random process happening, which based upon some previous activity, which creates associations and these are matched to the incoming input. So for that, you need to identify prediction errors, right? And so if there's no prediction error, you say, oh, this is what they're out there. This is what the stimulus out there. But if there's a prediction error, then it's different. But one can look at it in different ways. One simple way of doing that is to look at a paradigm like this, which was developed by Jason Mattingly and his colleagues in Brisbane here in Australia, where they have these blocks where there are two types of trials. One is and the first and the second are the same, or they could be alternating, two different orientations. But in different blocks, the percentage of these are different. In some blocks, they're repeating more. 80% will be the same, repeating blocks, and 20% of the trials will be alternating. Now, others alternating blocks, where most of them will be alternating, and repeating will be less. You can call them match and non-match. These are match trials, these are non-match ones, and they're similar to the monkey ones. So they had, they did EEG. EEG doesn't have, right? Oh, I want to show this first. Okay, the EEG, this is what I meant to show earlier. Okay, and then I was going to show this. Okay, now this one. And then they were able to decode from the EEG signal the degree of orientation selectivity of this system or the visual cortex in general, but as I said, EEGs, spatial localizations rather poor, but still what they could make out is the prediction error. That is, in those blocks where the non-match were unexpected, there's a higher response here. It's a kind of a prediction error. Okay. Now, since we could do the same, we did the same thing in the monkey. This is a very recent study. We have the two sorts of blocks, and in those blocks just in those blocks where the non-matched stimulus was unexpected, you get this very clear response here. These are stimuli, these are even blocks where the monkey doesn't even pay attention to that. It's just happening. They're paying attention to the fixation spot. And even when they don't pay much attention, the fact that some stimuli are more expected than others does make a significant difference. This kind of prediction errors will happen because in those blocks where you expect most of them to be match, and when the non-match stimulus appears, there is this error signal is there. All right. Now we have in fact done something. It's very hot. Just recorded just a couple of days ago, and today we're recording from not just from V1. This is V1. We have 3 electrodes: one in V1, one in the parietal cortex, one in the prefrontal cortex, and we find that the prefrontal cortex, you find this prediction even when the first stimulus is different. Already, you find the stimulus here, or you find a clear significantly different response here, which means that the system already knows this is a different one here. This is going to be this could be a non-matched stimulus, right? And so there could be this top-down activation. Higher areas are showing that earlier, and the lower areas actually match it to the stimulus and say, okay, here is an unexpected one. Okay, so this was predictive coding. Now, I suppose I should stop there. To give you I told you how feedback from the parietal cortex, from the LIP, modulates response in the V1, and it's mediated by the synchronized oscillations neural activity in a certain ranges, frequency ranges. And then I showed you briefly how there are 2 types of cells, one showing more attention enhancement but poor feature selectivity, and the other one other way around. Together, they've been mediating this top-down feedback, which helps highlight spatial locations of objects of interest, especially when you have a lot of objects that you do a serial process of looking at, retains memory of those locations, but at the same time, being able to throw your attention on one or the other. And you do find significant effects in V1, attentional effects, and also probably even predictive errors, even when the monkey is not paying so much attention. Okay, thank you. These are people whom I like to acknowledge. Shuri Katya, who done quite a bit of the recent analysis, and Yuri, Salman, and Ivan. They were ones with whom I recorded some of the earlier ones I showed you with the other study, looking at coherence between MT and LIP. And more recently, I've been working with Katya and Mojtaba Kermani and Yamini, and Jay has been working with us 2 years ago when he moved to Chennai. And the recent work, the predictive coding, I've been doing with Jason and Matthew from Queensland. Thank you. Hello.
Hello, sir. Thank you for your wonderful talk. If audience have any question, you can ask this, sir.
Okay, now can I see them? No. I'm not able to see anybody.
Does anyone have any questions? On behalf of our organizer, I would like to thank the sir for giving his valuable time for the session and for this talk. It was wonderful listening to you, sir. Thank you.
Thank you. Bye.